Welcome back, friends. We've been talking about the sexual reproduction in flower, and we have also talked about the different steps of the sexual reproduction in flower. And we divided this chapter into three different parts. Pre-fertilization event, where the plant become prepared for the fertilization. Then the event of double fertilization, which is unique to angiosperm or flowering plants. We have already seen that. So if you haven't seen that part, I will recommend you to go and watch that video in my channel, because that will make more sense to understand this video, which comes to the third part of our chapter, that is post-fertilization events. Post means after, pre means earlier. So post-fertilization event means events that occur after the double fertilization in the flowering plants. Now post-fertilization events always combined with several different changes that continue in the cell, because what we understood at the end of the double fertilization is that the pollen grain, or you can say the male gamete, start to take entry and fertilize the egg cell that is produced inside the ovary. So once the egg cell is fertilized in here inside the ovary, now that egg receives the nucleus from the gamete, male gamete. So now, as we know that both the gametes carry n number of chromosomes, so altogether it gets 2n number of chromosomes and turn into zygote. Now that zygote needs to divide, and that zygote will convert into an embryo. So once the embryo of a plant is formed, now that embryo can give rise to another future generation of that same plant species. That's the idea of sexual reproduction. Produce an offspring from an existing one with the help of fertilization. Now we've seen how fertilization take place, but the problem is, after the fertilization is done, that means we formed the zygote. The zygote is already ready. Once the zygote is ready, now it's job for the zygote to divide. What kind of division? Mitotic cell division. Mitosis, that means the zygote will become two cells, from 1 to 2, then 2 to 4, 4 to 8, and so on. It continues to divide and make huge number of cells together, and they start arranging themselves and make different type of shapes, and those different shapes will give a rise to the formation of plant embryo. And once the embryo is formed, then that embryo now turn into a specific structure which we are all very much familiar with, that is seed. So have you ever imagined what a seed really is? It is an embryo encapsulated by a coat, a hard coat, which protects the embryo inside. So whenever you carry a seed and implant it into a soil, that seed, that embryo that is present inside the seed, start to come out as a plant. That's how we see new plants coming in from seeds. That's the idea. So how exactly a zygote is turned into embryo, and then an embryo is making another whole new plant? There are two separate ways to that. The process of producing embryo from zygote is known as embryogenesis, and from that embryo, production of the full plant with the shoot as well as root is known as what? Germination of the seed. So seed germinate means it will start rising different part of the plant, shoot and root, and they'll start taking nutrients from the soil, and then the plants start to grow, because the cells start dividing once they start getting the nutrients from the soil. So here we are going to talk about the post-fertilization event, and three things that we want to talk about. The part of the seed and embryo, that is endosperm, embryo and seed. We are going to talk about their origin, their structures, and how they mature in the future times. Now remember one thing, I have told you the generation of endosperm in the last few lectures, that how exactly the endosperm is generated. Remember, during the fertilization in flowering plants, two fertilization events take place. In the first fertilization event, so let me write it here. In the first fertilization event, there is this male gamete fuse with the egg or female gamete. That is the term known as first important fertilization, and formation of zygote takes place. While the second round of fertilization also is there, and the second round of fertilization is another male gamete, because in flowering plants they produce two male gametes. The other male gamete will interact with the polar cells, and there are two such cells, because in polar cell means there are the two nucleus that are present, N plus N. So here is 2N nucleus that was present. Now the other gamete, the other male gamete with N number of chromosome fuse with the 2N number of nucleus or chromosome containing nucleus of polar cells, and now they will be converted to a 3N chromosome containing cell known as triploid cell. Triploid cell, and this triploid cell is actually known as the endosperm. And we know this triploid cell or the endosperm, which is also known as the primary endosperm, which is just produced right after the fertilization event. This primary endosperm is the nutrient source for the developing embryo. Now, where exactly the embryo is developing? Two separate events I just told you. Two fertilization events. That's why we call it double fertilization. The first fertilization produced the zygote, and the second fertilization event produces the endosperm. Now the endosperm provide the nutrient values to the zygote so the zygote can grow. So once a zygote is present and this endosperm continuously providing them the nutrients which is required for the cell to grow, and the zygote is now slowly maturing and turning into different types of shape and structure. So changing in structure and shape slowly once they start uptaking nutrients. Now the zygote is first converted to what is known as globular embryo, globular state embryo. Now zygote, one small modified right after the production of endosperm, is known as embryo, because there is no much difference between a zygote and embryo. The only difference is the zygote start dividing mitosis, cell division, by taking up nutrients from the endosperm. So zygote is converted to embryo only if there is a specific amount of endosperm produced in the cell. Otherwise, if there is no enough endosperm produced, zygote conversion to embryo will not happen. It's very, very important, because without endosperm there won't be any nutrients. So zygote conversion to the embryo will not be possible, because they need constant requirement of nutrients. So first they'll produce this globular state, and then this globular state is converted to heart state or heart shape. All this depends on the structure of the embryo. The globular state is something like a circular mass of cells and tissue that is formed from the zygote. That is the globular stage. Now the globular stage, the cells start dividing and arranging. They'll form a heart shaped structure. Once the heart shaped structure is formed, let me erase some part of this idea from here. So once this heart shaped structure is formed, then they'll convert it into a torpedo stage. It will look something like this, a torpedo stage, which is also a type of cylindrical structure in nature. So once torpedo stage is reached, now that is kind of at the end. And although in this picture I've drawn a specific arbitrary drawing, but we are going to see the structure of embryo in details, which is the matured structure of embryo. And you will see this matured structure of embryo, they have different types of structure, depends on what kind of plant that we're talking about. Mainly there are two types of embryo that you can see. An embryo with two separate components and parts of their body, which, because the job of an embryo is to create two separate sections. One section which will develop in shoot of the plant, the other section which will develop in root of the plant. Both these things are very much required to form, and they are always connected by a small set of tissue lining known as the embryo axis. Now the embryo axis has two parts. The upper one will make the shoot, the down and bottom one will make the root region of the plant. But for making this structure and different part of the plant, they need to have differences and different way of expressing and also managing their cell division. So we'll study that in the next part of the class. But now these are the conversion from zygote to globular, from globular to heart, from heart to torpedo, and from the torpedo they will produce this mature embryo. Many places you will also find that from heart directly they say the mature embryo, but actually torpedo is a kind of intermediate step between heart and mature phase, which is kind of an indication that now the embryo is almost mature, now it is enough and able to produce a new plant. So once the embryo will be produced, the endosperm that we are talking about at this point, the endosperm, the normal endosperm, the primary endosperm tissue converted to the secondary endosperm, or what we can say simply. Now meanwhile, while the growth of embryo that we are watching, remember, the growth of embryo and modification of zygote into embryo depends on the availability of the endosperm. But the endosperm itself is also modifying, it's not remaining the same, because embryo as it's developing taking up endosperm. So what will happen at the end of the embryogenesis? According to this idea, all the endosperm should be taken up by the zygote to convert into a mature embryo, but it's not happen all the time, because in few species of plants not all the endosperm are utilized to convert into a mature embryo. Many part of the endosperm already remains there, which we eat as good source and nutritious food that we eat. For example, coconut is an example, or barley or wheat. In all these cases, in all these plants, they have not taken complete endosperm during the embryogenesis. So they take few part, but most of the part remains, which we eat with delicious nature of all those fruits. Okay, that's the idea. On the other hand, there are few plants where the plants during the embryogenesis take complete endosperm. They haven't left any endosperm out there, take completely every part of the endosperm. Example for such, groundnut. Examples for such are pea plants, or any kind of bean type of plants. Those plants, they take complete endosperm and they utilize complete endosperm for the development of embryo from the zygote. That's one thing. But how the modification of endosperm take place? Let's look at that in a moment. So understand that process. I am deleting rest of this part which you've already talked about. So let me erase it and let's talk about the endosperm and the modification of endosperm. So remember, this is the endosperm. The endosperm that is formed from the primary endosperm tissue. Now that endosperm is nothing but a layer of tissue that is surrounding a part of this the zygote that is present in the middle. So it's continue to supply the nutrients. But this endosperm also modify, because what happens to the endosperm? It carries 3n number of chromosomes in it. So the nucleus in endosperm, 3n number of chromosomes. So this triploid cell, they also start dividing, and this division will produce many nucleus. Let's say nucleus 1, 2, 3, 4, and many nucleus in that same lining of the tissue. So one cell, multiple nucleus start to circulate in that place. Now what kind of situation is this? Because the division of nucleus is not followed by cytokinesis, that is a separation and formation of cell wall. So formation of cell wall is not occurring, only the replication of chromosomes and making new nucleus. Now at this point we call it as a what? Free nuclear endosperm. In short form, FNC. At the very beginning we call it primary endosperm, or simply PEN, primary endosperm, converted to free nuclear endosperm, where we have all the nucleus surrounding but without formation of any cell wall. Now not all this free nuclear endosperm will remain as it is. Now this free nuclear endosperm should be converted into another compact form by providing cell wall circulating each nucleus, and that is the end, because that particular form that they will form it will be known as the cellular endosperm. So what happens here? So let's say especially this whole event is very much clear. The development of endosperm is very clear in coconut. Remember one thing, in coconut what we see, that if this is the seed, this is the coconut seed, and what we can see is simply the outside structure, the seed coat is there, and now inside, let me draw it with different color. This is the white solid region of the coconut. Remember, the white solid region of the coconut, and you know that is endosperm, right? Because we eat it and we know that it is nutritious. This is the endosperm. But what about the water? From where the water comes from? It is also a part of endosperm. So now let me draw the water in the middle. This is also part of the endosperm. Now what happens during the modification of the coconut seed? This part, you know, the liquid part is nothing but free nuclear endosperm. Now this free nuclear endosperm will remain as it is because the nucleus is not completely covered by the cell wall. It's not created because the maturation may not take place completely always. So that thing may continue. On the other hand, the tissues that are circulating and surrounding that liquid region or the nuclear endosperm are the tissues where cell walls start to form. And as cell walls start to form here, that will become hard and solid because you know cell walls provide strength to the cell. So surrounding regions are very hard and the center part is liquid because the liquid part is free nuclear endosperm and the hard part is known as the cellular endosperm because they properly form cells, right? So this is cellular endosperm. Okay, so this is how the endosperm maturation takes place always, you know, and it also depends on few type of plant species who utilize most of the endosperm, which we've seen like for pea or beans. But for coconut or other examples like, you know, as I told you earlier regarding barley or wheat, in all these cases they haven't take the complete endosperm, they only take few part of the endosperm because that's why, you know, if you keep this seed for a long time and they start germinating, they are taking some part of this liquid endosperm and also part of this free cellular endosperm as well. So nuclear endosperm as well as cellular endosperm will be uptaken the more you keep the seed when they start to produce the structure of the embryo. Otherwise you'll see most of the time when we take the coconut, it is kept as it is because there haven't been a much embryogenesis happened there. The embryo is formed, it's in dormant state, so the embryo have not converted into a plant. The embryo is produced but it is in dormant state. That's the idea of seed. Why a seed is produced by a plant? Always the idea is simple, because once we produce the embryo, we need to protect the embryo. So we have a seed coat, a hard shell outside that will protect that seed against all the environmental problems. That is one thing. The second thing, because plants need to disperse its seed. If it only produces embryo which is cellular, it may not be beneficial for plants to supply them with air or water because it will not survive. But with proper measures, it can survive long time and a different range of complexity. Another important thing is that, you know, the embryo can be preserved for a long time and the embryo is ready. But what happens in seed is that most of the seeds, they remain dry because once the structure of the embryo, from at the end, they produce a small pore. And remember from where the pore is produced? Micropyle pore. You know, you remember we talked about the process of the gametogenesis, the male and female gametogenesis. I told you about the micropyle pore during the anatomy of the female gametogenesis process. And I told you that pore, that micropyle pore also remain open all the time. A small pore which helps the embryo to keep alive, to take oxygen and other important materials required from outside. But the water content in all this embryo is very less, like 10 to 15% of the natural water content. That's why they remain dry. They are not ready enough for the cells to divide and turn into a mature adult plant. So they remain what we call as a dormant state. Dormant means they're not ready to come up as an adult plant. Now what will happen if you take that embryo and place it in presence of proper water supply and heat, temperature, light? Then what will happen? The dormancy will break because they will sense the availability of water surrounding the seed, and then at that time they will take the water, and there is sort of chemical reaction always continue to happen inside this embryo that will allow the embryo to break that dormancy and start to divide the cells in the embryo. Because I told you it can carry two separate parts, division from the upper region is known as the epicotyle region, and the bottom one hypocotyle region. So hypocotyle and epicotyle, from both these sides they'll start dividing the cells. One part will produce a shoot, another part will produce a root. That's how the whole plant growth will begin. That's the whole idea. And this process of breaking the dormancy of a seed and generating a complete plant cell is known as the germination process, or process of germination. We'll also talk about that in details in the extra part of this class. But this is the event of how the embryo looks like, and how the components of embryo like the embryo as well as the endosperm, and how they develop simultaneously together. Now in the next part of this lecture, I'll talk about the structure of the embryo, and we'll talk about two types of embryo which is generally found: monocotyledonous and dicotyledonous. Now monocot and dicot are two separate types of plants depending upon the type of embryo that they produce. Although remember one simple thing, the first few stages of this embryogenesis is exactly the same between monocot and dicot plant. There's no much difference, because in both this case they utilize this conversion of pro-embryo into primary like the structures of the globular stage, then heart shape, torpedo stage. These steps are there, but you know how the structure will be arranged of the embryo is a little different in both this case. So their anatomy is little different, but the dormancy breaking process is also slight different. But the very first few stages of embryogenesis is very much similar between the two. That's why I said it as a common part, but specifically their anatomy and the growth pattern will be little different, which we'll see in the next part.